First of all, a very warm welcome from my side for our first webinar scientific session in 2024. It's really great that you all could make it in time to join this new, very interesting webinar session from today. So basically, let me then start to introduce first of all our speaker for today. This is Dr. Yassir Shuaib. He's a new team member responsible for our new assay, the tuberculosis LAM assay. Okay. And the topic for today's webinar is the PATHFAST TB LAM antigen test, a novel tool for TB treatment and monitoring. Okay. So as already mentioned, so Yassir joined our team just recently, just to care about exactly this assay only. So the position from Yassir is he's our business developer for the tuberculosis diagnostic assays, okay, at PHC. Okay, what about his objectives generally? So his objective is, of course, to promote the introduction and the complete success of the biomarker in the PATHFAST tuberculosis assay market, okay? Regarding his experience, he owns really extensive experience and expertise in the PATHFAST in the field of tuberculosis diagnostics and also molecular epidemiology. Okay, he did his PhD in Germany at the National Reference Center for Mycobacteria and molecular mycobacteriology mycobacteriology laboratory in Boston in Germany. Okay, he owns really deep understanding of mycobacteriology and molecular diagnostics. So this is why we are really happy and proud that he now joins our team. Okay. During his doctoral studies, he further enriched this knowledge at the WHO Supranational Reference Laboratory of Tuberculosis in Gauting, also in Germany, where he contributed significantly also to tuberculosis diagnostics and all activities around this, okay, and also quality assurance. Okay, generally, Yassir's professional career reflects a more global perspective with involvement in tuberculosis control programs across continents, including even Africa, in Sudan, and also Asia and Nepal. Okay. And he has really deep understanding of the tuberculosis diagnosis and monitoring, which is also coupled with a really broad experience in international organizations and reference laboratories of tuberculosis, which really positions him being an expert in this global field. Okay. So today, now Yassir will guide us through today's discussions on the introduction and impact of the PATHFAST tuberculosis LAM assay. Okay, so this is generally some words about our speaker for today. And so now I would say, please join me in welcoming Yassir, so Dr. Yassir Shuaib, to this very interesting webinar. Okay. So regarding the rules or the settings for this session for today, please feel free to state any questions you have during this scientific session. Please state all these questions in the Q&A section here. You can find this in the line on Teams. Please state every question there in the Q&A session. Okay, and we will come back to these questions after the meeting. Okay, so we will then have the occasion for this Q&A session after the meeting. Okay, please feel free to do this. All right. Okay, so Yassir, then I would like to hand over to you for your talk, and please enjoy.
Thank you very much, Frank, for the nice introduction. And as you already stated, today we will explore the PATHFAST TB LAM antigen test, which is a new diagnostic tool for monitoring TB treatment test. The test was developed by PHC in Japan, and we will see how can this novel tool can transform TB treatment and care. So I have divided the presentation into different parts. First, we will delve into the background of TB and TB treatment monitoring. Then we will see some clinical trial data that were generated in Japan using the PATHFAST TB LAM antigen test and speak about the potential applications of this test in clinical practice. We will conclude by summarizing the key points, and then we will have the Q&A and discussion session. So tuberculosis, or TB in short, is caused by Mycobacterium tuberculosis bacteria. It primarily affects the lungs, leading to pulmonary tuberculosis and causing a range of symptoms, including persistent coughing and chest pain, among other symptoms. One might ask how tuberculosis is transmitted. So basically, if you see here, we have one person who is infected with tuberculosis and another person who is healthy that does not have tuberculosis. What happens? The infected person releases the bacteria in tiny droplets in the air while this person is coughing, sneezing, or even when this person is speaking. The person in the near proximity of this infected person inhales the infected tiny droplets with mycobacterium, and those tiny droplets travel and settle down in the lungs. When they settle down in the lungs, they cause the infection. And one of two things happens. Like 5% of those people who inhale the tiny droplets, like 5% of them developed active tuberculosis with symptoms and become infectious within the first 2 years of inhaling these tiny droplets. And about 95% remain latently infected, meaning they show no symptoms and they are not infectious as their immune system is able to contain the Mycobacterium tuberculosis complex. And this is what is known as latent tuberculosis. But what happens over the course of their lives? 5 to 10% of them will develop active tuberculosis. The latent tuberculosis will become active tuberculosis. Also, there are a number of risk factors for tuberculosis, including like HIV infection, like young age, malnutrition, and diabetes, and others. So, as you can see from this map, tuberculosis, or TB, is a global problem affecting people in all parts of the world, with the highest burden of this disease concentrated in Africa and Asia, as you can see here. In the year 2022 alone, an estimated 10.6 million people fell ill with tuberculosis, and nearly 1.5 million people tragically lost their lives due to tuberculosis. This translates to roughly 28,000 new cases and around 4,000 deaths every single day. If you think about these numbers, they highlight the huge human cost due to tuberculosis. These are not simply numbers. They represent like a person, a family, and a community affected by this devastating disease. This disease, however, is treatable, though it leads to huge human cost. It can be treated. So how is this disease being diagnosed? The diagnosis of tuberculosis is comprised of a cycle is starting from clinical evaluation. The patient goes to the doctor. The doctor takes clinical history and makes clinical examination, and then the doctor establishes whether this person might be infected with tuberculosis. After that, the doctor sends the patient to the laboratory, and the patient gives samples, and the samples will be investigated with a number of laboratory tests, as we will see in the coming slide. And then the results, whether true positive or true negative, are reported back to the doctor, and the doctor, based on these results, decides the treatment. So confirmation of tuberculosis is done by a number of laboratory tests, as we said. These include bacteriological tests, molecular tests, biochemical or immunological tests. Examples for bacteriological tests include smear microscopy and solid or liquid culture. And examples for molecular tests include PCR, such as GeneXpert PCR. Also, some molecular tests like line probe assays, which are very useful for species identification and species differentiation, as well as drug susceptibility testings. Some laboratories with capacity use whole genome sequencing, or even use Sanger sequencing, or even whole genome sequencing. While these days there is a trend of using biochemical or immunological tests in specific groups of TB patients, such as lateral flow assays from Abbott and Fujifilms. Each of these tests has its own advantages and disadvantages. For example, smear microscopy is rapid, cheap, but it's less sensitive, while culture is the gold standard, high sensitive, but it takes long time before the results are available. Molecular tests are sensitive and can be used for drug resistance diagnosis. The lateral flow assays are also rapid, cheap, but less sensitive. So there are different purposes for the different groups of the TB tests, but each test plays a distinct role in patient management, as we see in this table. The intended use of TB tests could be for screening and initial diagnosis, or for DST or drug susceptibility testing and treatment monitoring, as well as to test for latent tuberculosis. For screening and initial diagnosis of tuberculosis, tests like smear microscopy and molecular tests and lateral flow assays are very useful, while for DST, culture and molecular tests play a very significant role. These tests utilize different biological samples like sputum, urine, blood, and give qualitative results, meaning they will just tell us if the patient is positive or negative. While some tests can give us like detailed information, and those tests are known as quantitative tests. They can give us like the amount or the quantity or the concentration of the target molecule. For example, in the case of the PATHFAST TB LAM test, these tests give us the concentration of the LAM. Culture can also give us a quantitative results, which is like the colony forming unit, but there are additional steps that we need to do to obtain this result. After diagnosis of tuberculosis, and when it's proved or confirmed the patient is suffering from this disease, there are several treatment regimens, several treatment regimes that can be recommended to the patient depending on many factors. One of these factors, for example, is whether the patient is having drug susceptible or drug resistant tuberculosis. The treatment for susceptible tuberculosis involves like 6 to 9 months, combining multiple medications like rifampicin, isoniazid, pyrazinamide, and ethambutol. As we see, the treatment is divided into two stages or two phases. We have the intensive phase and the continuation phase. The intensive phase is as long as 8 weeks, while the continuation phase could be 18 weeks or a little bit more or a little bit shorter than that. And you have to remember, like, monitoring treatment response is really very important during this lengthy period of treatment. So the doctors want to know if the treatment the patient is taking is actually working or not. Because if it's not working, we don't need to waste resources and also allow the patient to suffer a lot. So this is why there are a number of interventions or testing that needs to be done during the course of the treatment. As we see on this slide, these interventions or follow-up comprise of different clinical practices and different laboratory tests, and they can be repeated at different frequencies depending on the purpose. As you see here, sputum, smear, and culture are very important, and they are conducted at the beginning for initial diagnosis, somewhere during the intensive course, and at the end of the intensive course, and somewhere in the middle of the continuation phase. So this means the recommended is like 4 times of testing, and this could also be done more than 4 times as needed. But there is one important notification here. Sputum testing by using smear microscopy and culture has limitations, as we will see in the coming slide. So this table is comparing the advantages or disadvantages of using smear microscopy and culture as diagnostic tests for TB as well as for TB treatment monitoring. Smear microscopy has limited sensitivity or very variable sensitivity, but it takes very short time to conduct, and it needs simple infrastructure, not like culture, which is sensitive and it's also the gold standard, but it takes long time. This could vary between 2 weeks to several weeks, and it needs sophisticated infrastructure. And we also have to keep in our minds culture can be contaminated, and this is another challenge. So a very important question at this stage comes to our minds: how to address these challenges of culture and smear microscopy when we are using them to monitor TB treatment response. The PATHFAST TB LAM test can really play a very significant role because, as we see in this figure, the PATHFAST TB LAM is highly sensitive and takes short time, not like culture. several weeks, and not like smear microscopy, variable sensitivity. So the PATHFAST TB LAM test combines the high sensitivity and the short turnaround time, making it a very ideal test for TB treatment monitoring, addressing the limitations of using smear microscopy and culture. And going back to this figure, uh, which we already discussed about the cycle of the TB diagnosis, and we said when the result is positive, patients should receive treatment. When the result is negative, no need to give the patient any treatment. But when the patient is given a treatment, then the PATHFAST TB LAM comes into play, and it can be used to really help in understanding how the patient is progressing over the course of the treatment. So another important question might jump to our minds at this stage, like, what is this PATHFAST TB LAM antigen test? Someone might ask this question. Well, the PATHFAST TB LAM antigen test is a novel diagnostic tool designed for in vitro diagnostic use with the PATHFAST automated machine. This is a PATHFAST machine which we use to quantify the concentration of the LAM. So the PATHFAST TB LAM antigen test is intended to be used in monitoring the response of TB patients to TB treatment, and also in identifying patients who might not be responding to the treatment. Another important question might jump to our minds also at this stage. One might ask, what is LAM? So to understand what's LAM, or to answer this question, what's LAM, let's have a close look at the structure of the cell wall of the Mycobacterium tuberculosis. Mycobacterium tuberculosis has a rod shape, as you see here in this red circle, and it has really a very complex and unique thick cell wall. This cell wall has, like when we take like a slice from this cell wall and magnify it here, we see the cell wall is having really different layers. It has the plasma membrane and the peptidoglycan or the arabinoglycan and the mycomembrane and the capsule. Have a close look here in between those two layers, the layer of the plasma membrane and the peptidoglycan membrane. In between those two, we have what's known as LAM. And when we magnify the LAM, this is how it looks. It looks like this, and it has its own structure, and it's composed from different subunits. This LAM, or lipoarabinomannan, is antigenic, meaning the body, when the immune system detects LAM in our bodies, the immune system produces antibodies that can react with this with the LAM. Another important feature of this LAM is that it's found in several body fluids like urine. It can also be found in the blood, and certainly it's found also in the sputum. This is why these biological samples can be utilized to diagnose tuberculosis and also monitor the treatment response in TB patients. So how does the PATHFAST TB LAM work? The PATHFAST TB LAM has 4 steps or 4 processes Those are the processes which occur in the PATHFAST machine, which we saw like few seconds ago. The processes are comprised of four steps. The first one is the immune reaction, and then the separation, and then the enzymatic reaction, and the detection at the end. So during the immune reaction, we will mix the sample. As you see here, the sample has like the target molecule and other components, and we have like magnetic particles coated with antibodies, as well as antibodies against our the molecule of interest, which is LAM in this case. So we mix those together, and then the antibodies will detect the LAM and react with the LAM. And in the separation, or in the second phase, we use the maturation technology, where we have a magnetic bead, and this magnetic bead attracts the magnetic beads which are coated with antibodies, and then the ones which are not bound, they will be washed away. And in the succeeding phase, we will have an enzymatic reaction, and in this enzymatic reaction, light will be emitted due to this enzymatic reaction, and in the final phase, the amount of the emission of the light is measured at 461 nanometers, like the wavelengths of the emitted light is measured. So to summarize this, the PATHFAST TB LAM uses the magnetic bead technology, which is known as the maturation, and also it uses the cilia technology to measure the amount of the light emitted during the test. How should we conduct the PATHFAST TB LAM test? To conduct the PATHFAST TB LAM test, we first need to extract the LAM from the cell wall of the bacteria. As we have seen, the LAM is really embedded in the cell wall of the bacteria, and we need to release this LAM from the cell wall. To release it from the cell wall, we take like 200 microliters of the sputum, and we mix it with sodium hydroxide, and then we heat it for 20 minutes at 100 degrees centigrade, and then we mix it with phosphate buffer, and then we do centrifugation, and we take the supernatant, which should have the LAM extract. After the extract is ready, we take 100 microliter and we dispense it in the ready-to-use cartridge, and then the cartridges are placed, the rack holding the cartridges is placed inside the machine, and then we start we start the run, and in 17 minutes we will have the results ready. So after this test has been developed in Japan, it also underwent like rigorous evaluation. The test was evaluated, or the reaction of the test to 5 Mycobacterium tuberculosis species was evaluated. These species included Mycobacterium tuberculosis and Mycobacterium africanum, Mycobacterium bovis, Mycobacterium microti, and Mycobacterium penipedii. The reactivity of the test to 178 non-tuberculous mycobacteria was also tested. In addition to that, 34 microorganisms which are found normally in the buccal cavity and the upper respiratory system, as well as 27 chemicals and drugs, including anti-tuberculosis drugs and anti-HIV drugs and other chemicals. And finally, the test was evaluated using 100 clinical sputum samples from the FIND biobank. The results were as follows. So the test has really a remarkable sensitivity. As you can see, it's highly sensitive. The limit of detection was 6.67 picogram per ml, meaning it can detect a very low levels of LAM in samples. That's equivalent to as low as 18 bacteria. So if you have 18 mycobacterium tuberculosis per 1 ml, the test can correctly detect this number of mycobacterium tuberculosis. What makes it very useful for treatment monitoring, even in patients with very low colony forming units. The dilution linearity was also tested, and we found that the test really performs very well, and in a range of 10 to 50,000 picogram per ml. This is like really a very wide range of concentrations of LAM, and we also did not see any hook effect associated with high concentrations. So the assay also detected LAM in various Mycobacterium tuberculosis complex, meaning if a patient is infected with Mycobacterium tuberculosis or Mycobacterium bovis or Mycobacterium microti, Penipidii, or any one of the Mycobacterium tuberculosis complex, this patient can be, the response of this patient to treatment can be monitored using this test. And we will see this in the following slide. And we also found really a linear relationship between the concentration of the LAM and the colony forming unit. And 1 picogram per ml means or corresponds to 2.72 colony forming units per ml of Mycobacterium tuberculosis. Like if you have 1, then this means you have 2.7 bacteria per ml. If you have 2, you multiply by 2.7. If you have 100, this means you multiply, you have 272 bacteria per ml. So this figure here shows the relationship between LAM concentration and the colony forming unit. As you can see, this has been done for tuberculosis and Africanum and bovis and microti, and also Penipidii. So there is really a very strong correlation with the colony forming unit. When the number of the colony forming unit increases, the concentration of the LAM, which is measured by the machine, increases, meaning the machine is really correctly is able to identify LAM and also quantify its concentration. Regarding NTMs and other pathogens, the test showed cross-reactivity with like really around 80% to 85% of the slow and rapid growing NTMs. But with regards to like common bacteria found in the buccal cavity and the upper respiratory system, there was a very weak reactivity. Only 3 bacteria reacted with the test at a different concentration. These bacteria are Nocardia, 2 species of Nocardia, and 1 species of Seculmurella. And there was no any cross-reactivity was found with other common upper respiratory and oral cavity microorganisms. And this table here shows how the table is sensitive. As we said, we used clinical samples from the FIND biobank to obtain these numbers. Though the number of the samples is low, but you can see the test is highly sensitive in comparison with culture, with a sensitivity of 88.8% and a specificity of 100%. and overall agreement of 91%. These figures are also from a clinical study which was conducted in Japan. Some patients were recruited, and those patients were followed up during over the course of their treatment. And as you see here, we could see really a perfect correlation between the concentration of the lamp and the time to positivity. If you see here, after diagnosis and before taking the treatment, like after 2 weeks of taking the treatment, the time to positivity like really reduced a little bit, but the lamp concentration was somehow constant. But like after 1 month, the lamp concentration went down and the time to positivity also reduced. And this happened till after like 2 months or 8 weeks of treatment. And we can see this also here, the similar pattern on this figure and on this figure and on this figure. So the treatment actually continued, and the data I'm showing here is only showing like 2 months. So after the study is completed, we expect the drop to go to zero or close to zero. The concentration of the lamp is going to be like less than 10 picogram per ml. And also the time to positivity is supposed to be like really close to zero or zero. What are the benefits of using this test, this new novel test? This test is very rapid. You can obtain the result, a meaningful result that will enable you to take action in under 1 hour. So you need half an hour for lamp extraction and 17 minutes for quantification of the lamp. And then the test is highly accurate. As we have seen, it can really detect as a very low amount of mycobacterial tuberculosis per ml, as low as 18, and it's very specific, 100% specificity. It's very easy to use, like people with minimal training can use this test, and it does not require complex infrastructure like culture. For example, to conduct culture, you need to have a biosafety level 3. But to conduct this test, you just need a biosafety cabinet, which is placed in a biosafety level 1 or biosafety level 2, and you need a heating block or a water bath and a centrifuge. That's it. You don't need really a very complex infrastructure. What makes this test like really very suitable for near point of care or even point of care with a little bit of improvement. So in what clinical scenarios this test can be useful? For early detection of treatment response. As we said, we don't want to have like patients taking treatments and then those patients are suffering the side effects of those treatments, and at the same time we are wasting resources and the patients are not responding to treatment, what might also lead to development of resistances. Like the accessibility of this test in resource limited settings. We can really implement this test in the like last mile villages, like where like it's really very hard to have tuberculosis infrastructure, but this test can be operated there if we have like a power source, which could even be from the solar system. It will very well help us to monitor drug-resistant tuberculosis, and also we can use it after completion of treatment to see if the patient is going to have a relapse or not. And at this stage, I think I have 2 or 3 slides. So, and just to give you an overview about the PATHFAST machine. The PATHFAST machine is a compact and user-friendly immunoanalyzer, as you see here. Like this is the machine. It's compact and it has like really a lot of advantages. It uses the cilia and maturation technologies, and its advantages, or it stands out because it has a compact size and it doesn't need like it requires a minimal bench space. It has high sensitivity and gives you accurate results in less than 1 hour. We have all in one, meaning the cartridges are pre-filled with all the reagents needed to run the test, meaning after lamb extraction, there is no need for additional consumables. Everything is really provided in this cartridge. You don't need to have any additional laboratory consumables. And in addition to this, it's very simple to operate. You just need to pipette or aliquot the sample into the cartridge and then place the cartridge or the rack holding the cartridges in the machine, and then press start, and in 17 minutes you have a very accurate quantitative results. Another additional advantage is this machine can really work with a lot of biomarkers, like emergency biomarkers such as troponin, D-dimers, and myoglobin, and it can also work with like biomarkers of sepsis such as persepsin and PCT. And it also works with the biomarker we discussed today, which is the LAM. So in this case, we will have a sputum, we conduct the extraction, and then we measure the LAM concentration. And one important thing also to mention is that this test is already CE marked in Japan, meaning that it meets all the standards of the EU, and we already started the process of the WHO endorsement. This is the slide before the last one, just to give you like really a graphical summary or a graphical representation of how this technology is simple to perform. Number one, sputum is collected. Number two, LAM is extracted. Number three, LAM concentration is measured. And number four, you have like really a quantitative data which gives you a precise information regarding the treatment response. This is the last slide. So the take-home key messages are that this technology is really accurate, it's highly sensitive, it can detect very low concentrations of LAM, and it's user-friendly, no need for like extensive training, and it requires like really minimal biosafety precautions or minimal biosafety requirements. We don't need to have biosafety level 3 as we do in case of conducting tuberculosis culture. And I also want to mention that PHCC is dedicated to improving health through digital and precision technology. And I would like to thank you very much for your attention. And I'm really happy to have your questions and discussion points. Thank you very much for your attention and for your participation.
Super. Thank you so much, Yassir, for this excellent presentation. Really super comprehensive, very, very detailed, super introduction to the new assay. Thank you for this, first of all. Okay. Okay, so then as previously announced, I would like to start the Q&A session right now.
Yeah.
And yes, of course, please feel free at any time right now to state any more questions you have regarding the assay, its procedure, its use, etc., etc. Please feel free to add any kind of questions in the Q&A section here, please. Okay, but let me start now reviewing the already stated questions. Okay. All right. So Yassir,
Yes.
people like to know why it's maybe not so well applicable for initial diagnosis, you know, for a primary setting, you know, just to diagnose the tuberculosis assay, you know, first. Yeah.
Okay. So basically, since the test can monitor tuberculosis treatment, it can also uh, be used for initial diagnosis. But in this case, it depends on really the person or the laboratory who is intending to use it for this purpose, because uh, the price or the cost of the test is a little bit higher than the tests which are being used for initial diagnosis. But like technically, the test can be used for initial diagnosis.
Okay, super. Thank you. Thank you for this short explanation. Moving over to the next one. So this is regarding the sample material, no, which is in use for the assay. And the question is here for the sputum, no, being the sample material for this. And there's the question about the, let's call it availability for sputum. So it's stated here that sputum is often difficult to obtain, of course, especially in children, or in some cultures also, it's not really so convenient to provide sputum, right? And so that's why the question is here. If we see some, let's maybe call it negative impact or whatever, for the usage of the assay, or for instance, it is also stated here that after initial therapy, the sputum production can be also reduced significantly. That it might be maybe in some way difficult to obtain either sputum at all, or maybe not enough material for this. What do you think, Yassir? Is there an impact, and if so, what is your opinion?
This is really a very excellent question.
Indeed.
We know when the treatment is going very well and it's like really curing the disease, some patients might have problems producing sputum. This is why we have planned studies now to investigate other samples like urine, like blood or plasma or serum, and also stool, because pediatric tuberculosis patients often they cannot give sputum. So at this stage now, like we have data generated using sputum samples. This is why I spoke about sputum samples alone, but we have planned studies to really evaluate the usefulness of other biological samples like urine, stool, and blood or plasma or serum. And hopefully, like the use of these biological samples could also be somehow related to the the concentration of the lamb in these samples could also be related to the colony forming units, so that we can use them also to trace how the treatment is progressing.
Oh, super. Yeah, this is really, really good to know, because we have also such kind of questions or kind of requests for other biomarkers, like for instance with prisepsin in neonates. Maybe you know that also here could be great to have urine being the sample material instead of, for instance, blood from the heel of the baby. So we are quite familiar with this. But excellent then, Yassir, that you have this kind of projection for the audience here, that there is something going on regarding this. Okay, super. The next one then is in terms of cross-reactivity with the non-tuberculous mycobacteria. Okay, as you already explained, but here the question is also a good one. Do you think it has really an impact on the test specificity here? What do you think? Is there an impact if they detect also, or if there's a kind of cross-reactivity possible? What's the
Yes.
impact? Can you state something here maybe?
Yes. So when we have the sample of a patient who is taking treatment, and we expect, for example, we established the first concentration as, say, just for example, 100 picogram per ml, and then we expect this concentration to go down when we take the second sample. And when we take the third sample, we expect the concentration to go down. But for some reason, from the second to the third sample, instead of we see going down, the concentration of the lamb went up again. This, of course, could be because of the reactivity of NTM. Maybe the patient got infected with opportunistic NTM, or maybe the sample got contaminated with NTM, or maybe there is no good response to treatment due to development of resistance. So when we see the result is not as we expect, this means we will test for resistance and also for NTMs. So we need to rule out the presence of NTMs. If there is no NTM, then we will say, is this really resistant? Then we test for resistance, and then we obtain like really accurate answer about the cause of the increase of the lamb concentration or the cause of failing of the lamb concentration to go down.
Okay, super.
Just to summarize this, what I wanted to say here, like the lamb will tell us if the treatment is progressing well or not. Instead of obtaining this information in 1 to 2 weeks using culture, we will know this information today when we use this test. Because if you are using culture, you will know the patient is not responding well in 1 week to 2 weeks when you use liquid culture, and even longer when you use solid culture. So using the LAM test will reduce this amount of time from 2 weeks or more to 1 hour. And then what you are going to do when you know the treatment is not going well after detecting this with culture, instead of doing it in 10 days from today, you can do it today.
Okay, super. Yeah, no, as you explained, yes. So as always, time is super critical, no, especially in an early state or later stage. Then, so it's really very important to save time. No, very good. Okay, very good explanation. So yes, for the audience, please do state questions in the Q&A session here now. Let me check. I think there might be other questions stated here, but I cannot. No. Here maybe. Okay. Okay. Yeah, it's stated here in the Teams chat. So please do state this in the Q&A session. But here I find something. So what could be interesting are the antibodies. Okay. So yes, I don't know. Of course, this is a kind of maybe also confidential information, but it's stated here. Which antibodies did you use as capture and detection antibody? Can we state something here?
As you said, this is confidential. So at this stage now, we will not disclose the antibodies used, but maybe later on, we will be able to tell people which antibodies we are using in this test.
Okay, understood. So next one is here then, it's about the accurate quantifying, as the concentration of bacteria in sputum would be quite dependent on the quality of sputum, okay? Do you think the quality of sputum might have an impact or an effect on the final result. And more than that, it's also a question here. Yeah, of course, this question could be expected. How about lump of death bacteria, not viable? Could you maybe have a comment on this?
Yeah.
Also regarding viable, so living cells in comparison to death cells,
Okay.
and the expression and detection of the LAM structures,
Yeah,
epitopes.
these questions are really very useful.
Yes.
Are very useful and excellent questions. So the sputum quality can actually affect the final result. This is why the doctors have to give like really clear instructions for the patients on how the patients should give the samples. Yeah, and it also depends actually on which part of the sputum you are pipetting. Yeah, some might take maybe like the sputum which is on the top. One of other people might go really deep in the sputum container and take from the bottom. And it depends on like the homogeneity of the sample. The more homogeneous sample you have, the more accurate reading you will have at the end.
Okay,
Yeah.
Understood. Yeah. And you have this heat extraction. I think this is a quite tough treatment also, right? To get it quite, well homogenized, no? I suppose, no? Right?
This was the first question, and then the second question was about LAM in dead or in live bacteria.
Correct. Yes.
Like if so, like when the patient comes and diagnosis of tuberculosis is established, at this stage, the first sample is taken, and say we have found a concentration of LAM at 1,000 picogram per ml. This includes the living and the dead bacteria. And then the patient starts taking treatment. In 2 weeks, like doing the first follow-up, if the treatment is working well, the antibiotics should be really killing the bacteria or stopping the bacteria from multiplication. And this will lead to the destruction, to the decrease in the number of the bacteria. And the decreasing the number of the bacteria will automatically result in the reduction of the concentration of the LAM. So even if you have dead bacteria, the concentration should go down because the overall number of the bacteria in the sputum is going down due to the killing effect of the antibiotic because it's working. And this should also applies for the second follow-up. The second reading should be less than the first and the number 0 reading, regardless of if you have the dead bacteria in the sample or live bacteria in the sample. And also something important, this technology does not really differentiate between LAM coming from the dead or live or viable bacteria.
Okay, it's a very good statement. Okay, understood. So, are there any more questions here from the audience? Please do state them now in the Q&A sessions. Maybe in the meantime, Yassir, maybe I could also ask you something, maybe.
Okay.
I mean, probably you covered already partly or not, but it could be really interesting, really maybe to explain again. You mentioned that it's not really needed for a laboratory or a site where the test is performed. it's not really needed having a safety standard 3, you know, safety security level 3, which is quite high standard since, you know, now tuberculosis is really threatening. But could you maybe explain why this is not really needed?
Biosafety level 3 is really like not available in many parts of the world, especially where tuberculosis is highly prevalent. Yeah, and therefore performing culture in those areas is a little bit challenging. But performing PATHFAST TB LAM test will not be challenging because we have two steps, which are killing the bacteria, making the sample like really sterile. So in the beginning, we are mixing the sample with sodium hydroxide, and the sodium hydroxide is known to disrupt the amino acid bonds or the protein bonds, and this will lead to the killing of the bacteria. And then heating for 20 minutes at 100 degree centigrade is also another additional sterilization process. So after those two processes, then the sample becomes non-infectious, and then handling the sample outside biosafety cabinet becomes really very easy. So there is no need for biosafety cabinet. There is no need for biosafety level 3 because like the heating and the sodium hydroxide are killing the bacteria, not like in the culture we are cultivating the bacteria and letting it to propagate and divide, and then it's live. And when we open it in an environment where there is no ventilation system, and this live bacteria comes out from these cultures, it can really pose risk for the people who are working. But in our case, the bacteria are being killed, and the handling of the sputum is done under biosafety cabinet, as I mentioned. And this biosafety cabinet could be like really located in a biosafety level 1 or preferably biosafety level 2.
Okay, okay. So that's a clear explanation. Thank you, Yasser, for this. It means really this treatment really kills everything. So absolutely no threat regarding infections, etc. So okay.
Correct.
Thank you for explaining. Maybe one last question from my side. It's regarding the treatment. You know, maybe you covered already partly, but maybe you can emphasize a little again on this part, because I think this is really a super critical and super important part. How about decrease or increase of LAM in the test? Does this really reflect a successful or non-successful treatment or therapy? What is your experience here, Yasir? Can you please say something?
I think here we can go back to this figure here. This figure here. If you take patient number B005, patient 005, like this is at day 0, like before taking the treatment, and then after 2 weeks, there was really a little bit of reduction of the LAM, meaning the antibiotics already started killing the bacteria and reduced the colony forming units a little bit. Okay. And after 1 month, we can see really the concentration of the LAM decreased. And this was associated with also perfectly associated with time to positivity. And so on. After 2 months, we saw also the concentration was going down. So like the decreasing concentration of the LAM is indicating like good or successful treatment response. But unsuccessful treatment response will look like after we have taken this sample at week 4, and then week 8, instead of this going down to here, it will go up to here. Do you see my cursor here? Yeah, instead of the concentration going down to 1, it goes up again to 3. This means the concentration of LAM is increasing, and this means the bacteria is still multiplying in the body of the patient, and this means the treatment is not successful. And as I mentioned also, the concentration of the LAM is perfectly related with the bacterial load or colony forming unit. Every 1 picogram, like every 1 picogram per ml, translates to 2.72 colony forming unit per ml. So if we have 100 picogram per ml, this means we multiply 100 by 2.72. Then we will be having 272 bacteria per each ml.
Okay, super. Okay, clear now. Okay, good, very good. Okay, thank you, Yassir. So then I would propose, since time is progressing, so last chance then, please do state any more questions from your side now in the Q&A session, please put it there. If not, so do we have something more? No. Let me check one last time. 1 minute, please.
I saw in the
sorry. Yes, there is one last thing here. So, at what time intervals the test should be used after initiation of treatment? So, could you please tell me about I mean the period of time.
And also there was one person who raised his hand. I think Adam Tower or something like this. He raised his hand. Maybe he wants to ask a question. But to respond to this question, like at what intervals? So the intervals depend on the national guidelines. So if the national guidelines of the country, for example, recommended follow-up 2 weeks after starting the treatment and 1 month after the first follow-up, and then another month after. This depends on the guidelines. So we cannot tell the TB control program how much testing they should be doing. This depends on the guideline. There is a clear guideline coming from WHO, and then each TB control program adapt these guidelines to their own guidelines in their country. So this depends on the guidelines. But as we see, as in this I presented this figure. This figure is the recommendation is coming from for example, the New York Bureau of Tuberculosis Control. So the recommendation here is to do 3 follow-up using sputum, like you do it like during the intensive phase and at the end of the intensive phase, and 1 time during the continuation. Additionally, you can do it as many times as you need. If it's really needed, you can do it as many times as you need. But the recommended is 3 follow-ups in addition to the baseline.
Okay, clear. So we have one last question, but it's not stated in the Q&A session. It's from Mr. Tavor, Adam Tavor.
Okay.
His mic should be open. So maybe, Mr. Tavor, could you please state your question now verbally?
Okay.
Seems.
No, there are two questions in the Q&A.
More
Hello, do you hear
questions?
me, Mr. Yasin?
Yeah.
Thank you for your insightful introduction and presentation. I just asked a question, you didn't answer my question, so I raised my hand. Thank you.
No
Yes.
problem. We will
Go ahead.
answer your question, definitely.
Most of animals are resisting mycobacterial tuberculosis, as we know, but if this test is applicable for animal origin source, I ask this question. Thank you.
Currently, the test is applied only for human TB patients. Currently, only sputum samples are used for the test. But for animals, at the moment, it's not recommended to be used for tuberculosis in animals.
Thank you, Mr. Yasin.
You are welcome.
Okay, let me check one last time. I think this should be all questions then stated.
I saw.
The moment I have a problem with the Q&A here. Okay, quality. What quality control? What quality control measures are in place to ensure accurate and reliable results? So yeah, yes, this is our control for the
Yeah. We have quality control kits. So when the machine is installed, we have kits to perform quality control for the machine, and then we have quality control kits for every lot of kits. Like when you buy a new lot of kits, before you start using this lot of kits, you need to perform quality control and register those kits. In addition to that, it's recommended to perform quality control weekly, like every Monday or every Sunday. If your week is starting on Sunday or on Monday, you can, before you start using the machine, there is a kit that you test first, 2 samples, and then after that you will be sure the machine is performing well. There is really a very well set of quality control, and this is not a problem, the quality control.
Yeah, because it's also stated here, does it also detect drug-resistant strains? I don't know what this question is. It's a control. Yeah, I mean, yeah.
No, we don't detect the assay cannot detect drug resistance. It just tells you how the patient is responding to the treatment. Is the treatment is successful or not? Instead of having this information when you use culture 1 week or more than 1 week, you will have this information today. And instead of using smear microscopy, which has variable sensitivity, you can have the reading with the smear microscopy today because it's variable sensitivity. You get the wrong answer, and you make decisions based on the readings of the smear microscopy. But in 2 weeks or 1 month, you discover your reading or your reading based on smear microscopy was actually not correct. But using this test, you will have less time and get a correct reading today.
Okay, super. Yeah. Okay, I think then we covered everything here. Thank you, Yassir, again for.
I see some questions also here.
Yeah, it's this one, dead versus viable bacteria, but it's not really a question.
Okay, but Natasha is asking, what are the potential downsides of this assay? The potential downsides of this assay, currently the reactivity with NTMs, because when the test tells you the treatment is not successful due to non-decreasing or due to increasing LAM concentration, one thing you need to do is to check for NTMs. Are there NTMs in the sample or not? Yeah.
Okay, super. Okay, I think then we covered all questions, right? I do not see any.
I still see 2 or 3 questions. People are still in the Q&A. Also, Patricia is asking, but the killing of the bacteria would be literate more lamb at the beginning due to the killing and then reduce. I did not understand this question very well.
No, at the moment.
But I can comment on this point, maybe generally. So if the bacteria is dead, or if it's viable. If the bacteria is dead and its cell wall is intact, this bacteria still has LAM. And if it's viable, it still has LAM in its cell wall. What we do during the LAM extraction is release this LAM, which is embedded in the cell wall, whether the bacteria is dead or whether the bacteria is alive.
Okay, good.
Yeah.
Yeah, fine. So perfect. I think then we can close the Q&A session for today, right? And Yassir, again, thank you so much for your excellent presentation. No, super. And yeah, last but not least, please let me give a last recommendation. So do not miss to join our next scientific webinar. This will be about the topic anti-proBNP. Will be held by a scientist from Switzerland. It will be on the 3rd of April. So you already received an invitation for this. We will remind you again through different channels. So please do not miss to join this webinar in April, 3rd of April. Okay, so then, thank you very much to all of you for joining this session today. And yeah, again, Yassir, thank you so much for your presentation. And I wish you all then a very nice afternoon, evening. Okay. All right. So goodbye to all. Have a good day. Bye-bye.